Fructose-uric acid metabolism is the key to a unified theory of metabolic disease. The missing piece is understanding the functionality of polyol pathway as a glucose sensor. It's about the glucose, and we start right now. Hi, my name is Dr. Pete. I'm a PhD biochemist. You can think of me as a biochemical analyst, and I'm also a nutrition network health practitioner. I've been working in the field for 5 years now, and I've been studying what I believe is the central driver to metabolic disease, and that's what we're going to talk about today. The polyol pathway, with its connection of glycolysis to fructose-uric acid metabolism, with the mitochondria at the center of all of this, is probably, in my view, the most relevant biochemistry that exists in our area right now. And we need to understand a lot more about it if we actually want to solve the problem of this multitude of metabolic diseases. Now, as we start our discussion about this central biochemistry, which is fructose-uric acid metabolism, I want to preface the entire discussion with the idea that this is an evolutionarily conserved biochemical pathway which is integrated with glycolysis and its own connections to the mitochondria. And why this was important to humans from an evolutionarily important aspect is because when we take in certain macronutrients, and we're going to talk about that, in elevated quantities, we actually have a shift from one type of metabolism that in this discussion I'm going to call burning to another type of metabolism, which I'm going to call storage, that is complete with systemic inflammation, elevated fat synthesis along with glycogen and the storage of water, and the alteration of nitric oxide signaling, which alters our blood pressure. Dr. Richard Johnson has talked and written about this extensively, and I provide you there with 2 references. When we activate this metabolism, which from the point of view of evolution was important to our survival because it allowed humans to the capability to rapidly put on fat, store glycogen and water. It drove our foraging capabilities, hunger, thirst. It elevates insulin resistance because that is necessary to make sure that the nutrient load coming in gets to all the relevant tissues that need it. We have the elevation of the systemic inflammation because we want to activate or stimulate the immune system. And then we have the elevation in blood pressure in order to make sure that all of the nutrients get where they need to go. And this is a natural aspect of being human. The problem with today in 2023 is that this metabolic pathway, this what I'm going to call the red state, is being activated chronically, day in, day out, week after week, month after month, year after year. And this is how we get to the constellation of metabolic diseases, which I'm going to turn to now. We know when this core biochemistry is activated, that fructose-uric acid metabolism is the driver of not only gout, but also kidney disease, diabetes, obesity, fatty liver disease, Alzheimer's and dementia, cardiovascular disease, coronary artery disease, and also sugar and alcohol addiction. And we know this from the body of Johnson's work, which is over 700 publications based on cellular and molecular data, based on animal studies of a wide variety, and also clinical studies in humans. On this idealized slide, glycolysis is depicted in gray on the left part of the slide. The polyol pathway, with its connection to fructose-uric acid metabolism, is shown on the right side of the slide in red. Our genetics predict that the polyol pathway, with its connection to fructose-uric acid metabolism, is potentially available in all of our tissues and organs. Therefore, the functionality of these 2 pathways will depend on the nuanced function of a particular type of tissue. The main function of glycolysis under low glucose conditions, these would be conditions that are 5 millimolar intracellular glucose or less, which is about 90 mg per deciliter of glucose, is to produce substrate level ATP required for basic cellular function, and the mitochondrial substrates, pyruvate and lactate, which will then enter the mitochondria and be burned to produce ATP. Lactate is in fact the terminal product of glycolysis and is formed in a ratio of about 7:1 lactate to pyruvate. Under these low glucose conditions, fatty acids that are in the cytoplasm can enter into the mitochondria where they are burned by beta oxidation to produce ATP. This is a cellular state that I characterize as burning, and I will refer to it in this video as the white state. Now, as glucose rises, as it would during a standard American meal, and by the way, we are going to focus on the liver as our model system for this part of the discussion. And as the glucose rises, polyol pathway opens, and we start to push glucose into endogenous fructose production and open fructose-uric acid metabolism. As the fructose is formed, it is immediately phosphorylated, and fructose-uric acid metabolism lacks all of the speed bumps that you normally find in glycolysis. There are no feedback mechanisms that slow the progression. So fructose-uric acid metabolism opens. We have the sudden decline in cellular ATP along with phosphate. We have a sudden acute rise in uric acid, which is a signaling molecule. We activate de novo lipogenesis, fat synthesis, the production of glycogen, along with the storage of water. We have an elevation of systemic inflammation, activation of JNK1 along with IL-1 beta, and we have dysregulation of the nitric oxide pathway at the level of the liver, which downstream has its effects on the cardiovascular system, one of which is the elevation of blood pressure. Additional activators of fructose-uric acid metabolism, aside from the glucose issue and the polyol pathway, include exogenous fructose coming in with added sugar, and added sugars can include both glucose and fructose. We also have alcohol as a major or profound activator of fructose-uric acid metabolism, in addition to processed food and processed meat, which usually include glutamate, inosine monophosphate, adenosine monophosphate, in addition to salt and added sugar. And there is evidence to show the expression of these pathways in brain, adipose, muscle, intestine, liver, endothelium, the chondrocyte, which is the biologically important cell that works in our joints, heart, and pancreas. This slide demonstrates the type of literature that you now find in regard to the polyol pathway as a glucose sensor and indicator of a metabolic switch, a metabolic shift from burning to storage. The polyol pathway signals a shift from the white state to the red state, where the red state is characterized by fat storage, glycogen storage, the storage of water, systemic inflammation, and the alteration of nitric oxide regulation. I have made the assertion that under conditions of low glucose, under 5 millimolar glucose, which is approximately 90 mg per deciliter, that the white state dominates the situation. This is the state of burning. But when we have a meal, a standard American meal that is high in glucose, and we shift the intracellular glucose above 5 millimolar or above 90 mg per deciliter, that we open the polyol pathway, which funnels into and opens fructose-uric acid metabolism. I've made that assertion. So now let's take a look on this next slide and talk about the key enzymes that are involved in this intimate relationship, fork in the road, if you will, or this shift in state from burning to storage. So under low glucose concentrations, that is to say intracellular glucose that's under 5 millimolar, the dominant enzyme is a hexokinase, I believe, which is called hexokinase 1, and it is active under conditions of low glucose. But once we have a meal that involves the elevation of glucose above 5 millimolar, then we activate an enzyme that is otherwise known as glucokinase or hexokinase 4. Glucokinase is activated over a range of glucose concentrations between 5 and 8 millimolar or 90 and 144 mg per deciliter. Now what I'd like to do is draw your attention to number 4. This is the enzyme aldose reductase. It is the first and rate-limiting enzyme in the two-step polyol process, and you can see becoming activated under the same glucose concentrations as glucokinase. Shifting back to number 2, you can see that glucokinase is also activated by micromolar amounts of fructose and sorbitol. And sorbitol is the intermediate product in the two-step mechanism of the polyol pathway that drives the opening of fructose-uric acid metabolism with the formation of endogenous fructose. Note that it takes 1,000-fold less fructose and sorbitol to activate glucokinase than it does the millimolar range of glucose required to activate this enzyme. Also, that the major product, glucokinase, driving glucose to glucose 6-phosphate, feeds back and serves to further activate aldose reductase. Lastly, shifting our focus to number 3, we see under the glucose conditions approximately 5 millimolar intracellular glucose required for glucokinase regulatory protein to release glucokinase so it can move into the cytoplasm to drive glucose to glucose 6-phosphate. That release of glucokinase at the level of the nucleus only requires micromolar amounts of sorbitol produced in the polyol pathway, and the first product of fructose-uric acid metabolism, fructose 1-phosphate. The take-home message is that there is a synergistic relationship between the glucose sensing functions of glucokinase, aldose reductase, and fructose-uric acid metabolism, driving the opening of the switch to the red state. So what are the activators of the polyol pathway? Shown on this slide, we see that hyperglycemia, alcohol, salt, dehydration, hypoxia, glucose 6-phosphate, NADPH, and urate all function to activate polyol pathway. Moreover, evidence has shown that the polyol pathway, along with aldose reductase, is active in the following tissues: pancreas, liver, heart and muscle, brain, adipose tissue, and including the intestine and also the endothelial layer of the cardiovascular system. Additionally, and with reference to the pancreas, this 2019 paper showed that fructose exposure caused beta cells to be hyperactive towards glucose and therefore influenced insulin secretion. On this slide, I am summarizing the biochemistry of fructose-uric acid metabolism in the human liver. Obviously, there are nuances in regard to this biochemistry in other tissues. Not everything is the same. For example, in adipose tissue, fructose, either produced endogenously or entering the adipose tissue from the outside, is converted directly to fructose 6-phosphate rather than being phosphorylated into fructose 1-phosphate. Under high glucose, glucokinase and aldose reductase are activated and glucose 6-phosphate begins to accumulate, leading to the activation of glycogen synthesis, and the polyol pathway is activated, producing fructose, which in the liver is immediately phosphorylated to fructose 1-phosphate. Once activated, fructose-uric acid metabolism cooks right along at 10 times the rate of glycolysis. Subsequently, the acute rise in uric acid causes the translocation of NADPH oxidase, or otherwise known as NOX4, to the mitochondria to inhibit the enzyme aconitase, which is required to convert citrate to isocitrate in the TCA cycle. Inhibition of aconitase results in the accumulation of citrate, which is then shipped into the cytoplasm and is a key substrate in de novo lipogenesis. In addition to an insulin effect, fructose-uric acid metabolism, once activated, induces the production of the transcription factors, carbohydrate element binding protein, and also sterol regulatory element binding protein 1C. These two transcription factors translocate into the nucleus of the cell where they activate the genes of de novo lipogenesis. including ATP citrate lyase, acetyl-CoA carboxylase, fatty acid synthetase, and also stearoyl-CoA desaturase. This is depicted in my diagram down on the right. At this point, the liver is turned into a factory for the production of fat. This results in the formation of oil droplets right in the liver and also the packaging of triglycerides into VLDL that are then shipped out into the circulatory system. Additionally, one of the main products of de novo lipogenesis, or the process of de novo lipogenesis, malonyl-CoA, feeds back and inhibits CPT1, which prevents the entry of fatty acid CoAs or acyl-CoAs into the mitochondria, where normally they would be degraded by beta oxidation to produce ATP. More on this later. It's also important to point out that this metabolism causes an alteration in signaling of the insulin receptor substrates 1 and 2 and functions to decrease the density of the insulin receptor on the plasma membrane of the liver. This can result in hyperinsulinemia and insulin resistance downstream in the peripheral tissues, in addition to hepatic insulin resistance. Remember that the fructose-uric acid metabolism at this point is rocking along at about 10 times the rate of glycolysis. One of the consequences of that is we end up pooling dihydroxyacetone, phosphate, and glyceraldehyde, which then feed back into the lower part of glycolysis. This is a Warburg or Warburg-like effect, consequences of which two things. The first is it provides a substrate level source of ATP that has been shown in the literature to be adequate enough to run the basic cellular processes that are required during activation of the red state. And secondly, this results in an overproduction of lactate. Remember, under burning conditions or the white state, that the normal proportionality of lactate to pyruvate is about 7 to 10 to 1. In this case, when fructose-uric acid metabolism is activated, that proportionality changes 100 to 1, lactate to pyruvate. The consequences of this shift is that we have lactate that is pushed into the circulatory system, which then works synergistically with insulin at the level of the adipose to downregulate lipolysis. Secondly, because of the added concentration of lactate within the liver, the lactate as a signaling molecule feeds back on both CPT1 and CPT2 along with malonyl to prevent access of the acyl-CoAs, fatty acid CoAs, to the mitochondria, where under burning conditions, those fatty acids would be degraded by beta oxidation to produce ATP. Now, after being in the weeds for a while, we are going to shift into a mode where we're going to summarize the effects of the regulators and the other components, the substrates that are involved in the activation of this shift from the white state to the red state. First, due to the activation of this metabolism and the acute rise in uric acid and its signaling effects, we see the inhibition of the cytoplasmic enzyme creatine kinase, which is involved in recycling ADP to ATP. We see the downregulation of aconitase due to the translocation of NOX4 into the mitochondria. Aconitase being a crucial enzyme in the TCA cycle, causing the backup of citrate, which is then shifted into the cytoplasm and is the key substrate in de novo lipogenesis. We have the downregulation due to uric acid of enoyl-CoA hydratase, which is an enzyme required for beta oxidation. Malonyl-CoA and lactate work synergistically to downregulate both CPT1 and CPT2, preventing the entry of fatty acid CoAs, acyl CoAs into the mitochondria. We have the activation of carbohydrate regulatory element binding protein and sterol regulatory element binding protein 1C that function at the least of it on activating de novo lipogenesis through the transcription of the key enzymes that are required for that pathway. And lastly, we have the induction of JNK1 and IL-1 beta that drives systemic inflammation. And not shown here is the alteration of nitric oxide signaling in the liver. This slide just reiterates one of the primary functions of carbohydrate regulatory element binding protein and the sterol regulatory element binding protein 1C with its effect on de novo lipogenesis. This slide just emphasizes the general features when carbohydrate regulatory element binding protein is expressed along with its homologs like Mondo A across various types of tissues. These effects include the transcription of GLUT5, the primary transporter for fructose, the transcription of fructokinase, the primary enzyme that drives fructose to fructose 1-phosphate, de novo lipogenesis, and also insulin resistance. On this next slide, we are going to summarize the relationship between the fructose-uric acid metabolism and its activation and insulin. So one of the key pieces of knowledge going into the summary of the effects of fructose-uric acid on insulin is to understand that up to 80% of the insulin that reaches the liver is going to be extracted. In other words, it's going to be pulled out of the portal blood into the liver and then degraded. And upwards of about 50% of the incoming glucose is also going to be removed. So anything that happens in the liver that alters this relationship will have an effect downstream on the amount of insulin that is going to reach the peripheral tissues and also the amount of glucose that's going to get there. And so with the liver and fructose-uric acid metabolism, we both have a hyperinsulinemic effect and also a downstream insulin resistance effect. We know this to be true because the insulin receptor density is decreased at the level of the plasma membrane of the liver during the activation of fructose-uric acid metabolism. We also see a decrease in IRS2 with an activation of FOXO1, followed by the induction hepatic insulin resistance. Keep in mind that if we reduce the insulin receptor at the level of the plasma membrane, then more insulin is going to pass by the liver and be pushed into the peripheral circulation, which means that we're generating a hyperinsulinemic condition and drives insulin resistance. Moreover, we know that insulin itself downregulates lipolysis at the level of the adipose tissue. This slide is to remind you that urate activates aldose reductase. It inhibits creatine kinase. It's involved in the translocation of NOx4 into the mitochondria where aconitase is inhibited, basically flipping the switch towards de novo lipogenesis. Urate also downregulates fatty acid oxidation by inhibiting enoyl-CoA hydratase and modulating AMPK activity. And lastly, urate upregulates JNK1 and IL-1 beta, driving systemic inflammation. It is important to recognize that urate is a signaling molecule that is involved in oxidation reduction signaling networks that serve to trigger or shift metabolic states. I summarize the data for urate here. Urate is an oxidation reduction substance that falls in the middle of a list of biologically important oxidation and reduction potentials. What this means is that urate can easily accept or give up electrons. In biological systems, urate tends to be a versatile electron donor. It readily forms urate radicals, and urate radicals readily form adducts with proteins. One final thing to say about urate is the fact that it elicits its oxidative effects, whether or not it's produced directly inside the tissue or whether or not it diffuses from the circulatory system into the tissue. Now we shift to another key regulator of the red state, lactate. Lactate inhibits lipolysis at the level of the adipose tissue. It decreases the concentration of GLUT4 at the level of muscle. It downregulates CPT1 and CPT2, altering the ability of fatty acid CoAs to get into the mitochondria. It alters cardiolipin, an important phospholipid which influences the functionality of oxyphos. It alters ATP production by the elevation of reactive oxygen species. And finally, downregulates fatty acid oxidation in the mitochondria. We've come a full circle. Remember what I said at the beginning of this presentation, and that was that I believe that the red state is perfectly normal for humans. It makes perfect sense when you consider the history of humans in regard to hunter-gatherers who were living under prehistoric conditions. The ability to shift to the red state allowed us to very rapidly put on fat, glycogen, and water to elevate systemic inflammation, to stimulate our immune system, and to alter the regulation of our nitric oxide system in order to elevate blood pressure. However, we were only meant to be in the red state intermittently, not day in and day out. The chronic red state is the reason why so many of us are metabolically sick. And finally, we come to the very end of it. So activation of the polyol pathway, along with activation of fructose-uric acid metabolism, is a metabolic switch. Once we throw that switch, we move to the red state, which is characterized by de novo lipogenesis, systemic inflammation, alteration of nitric oxide signaling. There's insulin resistance along with leptin resistance. We didn't talk about leptin, but the leptin aspect of it is also there. One thing I didn't address in this presentation at all was the fact that there are behavioral aspects to this that have also been shown in the literature. Red state activates hunger, the over intake of food. It has an activation of the dopamine reward system in the brain. And there are also hormonal aspects of this, like delta phos-B, which drives the need to go back to the substances that are favored by the red state. So that would be sugar and fructose as an example. Also, vasopressin is expressed during the red state. And then finally, the disease platform that comes out of this. Again, we all have individual genetics. Then we have the food environment that's interacting with those genetics, along with other aspects of what it means to be human. And that interaction can result through activation of the red state in chronic diseases like diabetes, obesity, and so on. I also think that this model lends credence to the carbohydrate insulin model of obesity, and it also supports the lipid energy model. The main reason is because the red state would drive both composition and proportionality of the different lipid particles. I would like to thank you guys today for watching my presentation. I know it was super dense, but this is something that I have worked on for a really, really long time, and I finally got it done, and I'm so thankful for that. So thanks for watching, and I will see you again next time.